Hello, good morning everyone, and I welcome you all for attending this seminar, and a big thanks to all of you for sparing your valuable time. And I'm pleased to introduce Ms. P. Sameera Sastry, who is one of the Ph.D. scholar, and she has done her research on induction of double haploids in chickpea and pearl millets, which is really a burning topic nowadays. She has done her M.Tech in biotechnology from Satyabhama University, Chennai, and B.Tech in biotechnology again from Malla Reddy Engineering College, which is affiliated to JNTU, Hyderabad. So now I invite Ms. Sameera to present her research findings, which you will enjoy, I hope, a lot. Please, Sameera.
Thank you, ma'am, for the introduction. And good morning to everybody and esteemed scientists. I'm really glad to have you all here. As ma'am introduced, I have worked on induction of double haploids in chickpea and pearl millet. And to just go beyond anther culture experiments, I have also gone for interolog mapping using protein-protein interactions. Yes. Yeah, so initially when I started my experiments, I wasn't planning on taking up pearl millet. It was only a focus on chickpea. So my objectives are as follows. Like in the first attempt was to reproduce the recent protocol for double haploids in chickpea by Girewal, which was published in 2009. And the protocol emphasized on stress pre-treatments for induction of androgenesis in chickpea. So it was an effort to reproduce the protocol, test its validity and reproducibility, and further try to improve the pre-treatment conditions so that we could get a better response. And the next is, in this same way, I have tried to give a preliminary attempt to anther culture experiments in pearl millet also. And next, to follow it up, we wanted to understand what is happening exactly in chickpea and pearl millet in relation to androgenesis. So I have gone for protein-protein interactions based on EST-derived interolog mapping. And I have also tried to employ some proteomics works, that is going for two-dimensional electrophoresis, in order to identify potential markers or triggers for androgenesis in these two crops. So I would not go into the details of double haploids as everybody is aware of them, but as a brief introduction, haploids are any cell or organism which has gametic number of chromosomes, and you get double haploids by simply doubling the chromosomes, which could be either natural, that is by endomitosis, or by chemically induced methods, say using colchicine. But double haploids in nature occur in very low frequencies, and they are usually abnormal phenomena. So they are not much of a use to breeding methods in natural conditions. So the first natural double haploidy was observed in Datura in 1920s. And later the in vitro protocols have been developed for Datura and tobacco and wheat, the first successful protocols. And as of now, we have more than 200 protocols for double haploids in many crops, and especially belonging to Solanaceae and Graminaceae families, which are the most successful crops for double haploids. And double haploids can be introduced to various routes, say androgenesis, parthenogenesis, apogamy, and white crosses. For my work, I was interested in androgenesis. So why double haploids? So I'm talking about double haploids, but why go for them? They're the fastest route to homozygosity. And you could achieve double haploids in a single laboratory-based generation, which would otherwise take some 6 to 7 generations in a field in a conventional breeding method. And for self-pollinated crop like chickpea, it is a very good means to represent a pure line or a new variety. In case of cross-pollinated crops, it would be used as a parent line for further hybrid generations. And double haploids are also superior to recombinant inbred lines because most of the RALs do not retain the homozygosity in spite of many generations of selfing. And double haploids, along with marker-assisted selection and express sequence tags, is the shortcut to plant breeding, and you could improve a lot of elite lines. And I think it is the best strategy among the best strategies to increase food production. in the next 40 years. So androgenesis. Androgenesis basically is the development of a plant from the male gamete. So that would obviously represent the donor, male donor. And anther culture and microspore culture is the most successfully used method for induction of androgenesis. And as I already mentioned, Solanaceae, Cruciferaceae and Graminaceae families are the most widely used plants and successful plants for androgenesis. However, legumes are still known to be recalcitrant, and even some woody plants are recalcitrant, and surprisingly, the model crop Arabidopsis thaliana, it is also still under experimentation for double haploids, but there are only preliminary experiments to test for the genes which are involved in androgenesis, but there is no protocol as such. The major disadvantage of androgenesis is the genotype dependency, and even among the species of a same crop, there is a lot of genotype dependency, which I have also observed in my experiments with chickpea. So this is a brief picture of the roots for androgenesis. The first pointer, this is the uninucleate microspore stage, and in the first pathway, it is having a equal division of nuclei, which is further dividing to give an embryo. In the second and third pathways, either one of the two nuclei, that is either the vegetative cell or the generative cell, is dividing further to give an embryo. In fourth pathway, both of them divide. In the fifth, you can observe that both of them divide, but there is a uniform division in one half of the embryo and unequal walls or irregular walls in the second half, which is called the endosperm domain, which further disintegrates, and only the regular walled embryo domain further develops. Another possible route could be after fertilization, but without fusion with the egg cell, where the egg cell disintegrates and we get an embryo, multicellular or embryo or a proembryo. So just to give a brief introduction of factors which affect androgenesis, the switch from microsporogenesis to androgenesis is a crucial step, and the switch basically determines whether we get a successful protocol or not for further inductions or not. So induction of microspores and the further commitment to the microspore embryogenesis is very essential, and it's a preliminary step to further development of callus and the plant regeneration. So the most important factors which would affect androgenesis are the conditions of donor plants. And it's usually observed that cereals, when grown in phytotron in controlled environment, respond better than the field selected crops. And as we already have seen, genotype dependency is very high in case of anther and microspore culture experiments, especially in legumes. And stage of microspore, mostly the uninucleate, mid to late uninucleate stage is ideal for induction of androgenesis. Further, some of the cereals, they even respond when a bicellular stage is cultured. It was observed, but for my experiments, I have selected uninucleate stage, mid to late uninucleate stage, which was ideal for me. And media also plays a very important role in induction process. In chickpea, we have observed that it requires multiple media at different stages of culture, instead of one single medium which would support both induction and embryogenesis, but chickpea seems to be complicated, it requires many media. However, pearl millet was quite simpler. One medium was enough to give us induction and embryos also. And stress pre-treatments, cold treatment being the most popular. It's usually given at 4 degrees for a period of 3 to 7 days. But in case of millets or in case of cereals, usually heat shock is preferred method. And in the recent times, there are many other unique stresses which have been used, say centrifugation, electroporation, even sonication for that matter, and irradiation also. So coming to chickpea, as you all know, it's the second most widely grown legume, and we have two varieties, Desi and Kabuli, and it's predominantly self-pollinated with a very low outcrossing level. So what is the status of double haploids in chickpea? There are protocols in legumes for lupins, for glycine and for pisum, but none of these protocols have been validated. Their reproducibility has not been tested, and it is not a routine tool in any of the pasture or grain legumes. though there are protocols, they are not used in breeding programs as of now. And the protocols are usually marred by very low induction rates and very little plant regeneration. And it's got a high genotype dependency and even seasonal dependency. Usually in the cool season, the buds selected in the cool season, they usually tend to respond better than in the summer. And as we already have seen, as I already mentioned, media requirements are very complicated for chickpea. And pyrimidine abiotic stress treatments have also been employed for chickpea. So the method I have followed, I have used 4 cultivars, ICC 4958, WR315, and ICCV 95423, and RID, which we have available here. And at uninucleate stage, I have selected the buds around 2 to 3 mm size, checked with astrocarmine staining of the stage of microspores, and then cold treated the buds for 2 to 4 days at 4 degrees centigrade. Next step is sterilization using buffered bleach for 20 minutes. The buffered bleach I mean to say here is sodium hypochlorite with H2PO4. So it usually doesn't give a shock to the anthers, so it's like a buffering. And then I have gone for centrifugation and electroporation, further followed by high osmotic media, which is having a sucrose concentration of around 17 to 80%. So centrifugation, we have, as per the protocol, the best centrifugation treatment is at 671 G for 3 minutes, but we have tried with starting from 10 G to 1000 G for a period of 3 to 10 minutes, 3 to 15 minutes. And electroporation, we have given 125 volts to 250 volts at 10 seconds interval and 3 pulses, 3 exponentially decaying pulses. After these 2 treatments, I have given an osmotic shock of like 17% sucrose or 8.89% sucrose. The stress treatments were given alone and as a combined treatment to check whether each treatment is single treatment is more effective or whether a combined treatment is more effective to induce androgenesis. So as per the culture media, I have used 12 different culture media. The base was the protocol followed by Greva, the CH media, and the hormones have been modified accordingly to suit our results. So I have the basic protocol is a CH1 media, which the protocol insists on. Further, we have changed the protocols with different combinations of auxins and cytokinins. And another 2 media from Garwal protocol, these RMD and RMIQ. So coming to the results, I have just given us, this is the effect of stage of culture. So as you can see, this is a very early stage microspore which was cultured, and most of the time early stage cultures have resulted in clusters. We do not know whether they are actually responding cultures or they are just clusters of early stage microspores. So I would say this is inconclusive and I would not consider it as androgenic response. But when late stage microspores are cultured, they have resulted in pollen grains. This is a mature pollen grains. But at mid to late uninucleate, you can see that there is one big microspore which is enlarged and which is induced. These are the non-induced microspores, but this one is an induced microspore, which is the ideal stage for culture. So coming to the centrifugation treatment, I have observed that 2 of the genotypes, ICC 4958 and WR315, they responded at 671 G, again resonating the protocol, but they also responded at a higher centrifugation at 1000 G for 3 minutes. Unfortunately, the RIT variety hasn't responded at all, and it's the least responsive among the 4 genotypes. And an observation is the Desi varieties respond in RMD media, whereas the Kabuli varieties respond in RMIK media. However, when the picloram and BAP medium was used, there was no need of centrifugation. Maybe that is because picloram is a superior variety of auxin than 2,4-D. It's reported to be very efficient in inducing androgenesis compared to 2,4-D. So when we have used picloram and BAP for cultures, we didn't need centrifugation at all. So this is the effect of electroporation, and these are fluorescent stain microspores using DAPI. And this is at 125 volts, this is at 150 volts, and this is at 200 volts. You can see the nuclei dividing here. That is the equal division of nuclei. And here also you can see the divisions. This is one nuclei and this is another one. But in 200 volts, there is no division in the nuclei. And further, most of the microspores have been dead. The viability is very low when the voltage is beyond 175 volts. These are another DAPI stained photographs of microspores. This is, you can see the cellularization here. It is the ICC 4958 variety, which is a Desi variety, cultured in RMD media. And this is 95423, a Kabuli variety, cultured in RMIK media. These were subjected to centrifugation and cold treatment for 3 days. And combination of cold and centrifugation was the best response that we have got. And here you can see the dividing microspores. It's a 3 nucleate microspore, and here you can see 2 vegetative, this is not clear, but this is 2 vegetative nuclei, and these are 3 generative nuclei which have divided. And this is a 4-celled 1, 2, 3, and there's another here at the other side of the wall. So the 4-celled microspore. And this is an 8-celled microspore. Yeah, it is squashed because of my squashing. But anyway, this is a multicellular microspore. This was obtained after 30 days in culture. And these were obtained after 10 days in culture. And these 2 were obtained after 25 days in culture. And as I've mentioned earlier, CHPB media, that is picloram and BAP, has given us the best response of multicellular microspores. As far as the media is concerned, again here picloram and BAP is the best response among the 3 genotypes. And the next would be dicamba and thidiazuron and RMD media. And these CHPB has responded for almost 3 of the genotypes. So I think that would be the best media to be used. So maximum response I have got is around 6.1%, which is not very significant. I believe it's quite low. And combination of pre-treatments is essential. And the picloram media with high percentage of sucrose, 8.8%, was the best among all the media tested. So we have seen Anther culture, but we wanted to know why chickpea has got such a low response, and it would also help us to understand the genetic basis of androgenesis. Since 2004, I think there are a lot of papers which have done transcriptome and proteome studies in androgenesis in model crops, and they have come up with a lot of genes which are expressed differentially during androgenesis and microspore embryogenesis. So there are 3 main categories where the genes are expressed. It is the cellular response to stress, and it is acquisition of the embryogenesis fate and suppression of gametophytic program. And the genes which are expressed have been classified under these categories. And here you can see the different genes which are being expressed, which have been studied. This is SERK, somatic embryogenesis receptor kinase, and AGL15. These 2 have been reported to be very essential for induction, and somatic embryogenesis receptor is capable of inducing embryogenesis even in somatic cells, even in non-gametic cells. And baby boom, this is BBM, baby boom gene, LEC1, LEC2. These genes, the ectopic expression of baby boom alone is enough to trigger embryogenic response. And during the plant regeneration and callousing stage, 1433A and AGP, arabinogalactans. A very interesting point about arabinogalactans is not only the expression inside the cells, but also externally. If we give arabinogalactans in the media, it has definitely helped responses in many plants. And though I haven't mentioned in my results, I would like to talk about it because I have used it in pearl millet experiments and it was found to be effective. So what I wanted to do for the genetic study, study basis of androgenesis, I didn't want to go for isolating cDNAs and going for transcriptome studies. It would be too much of expenses and work for me. So I first wanted to test for in silico expression of genes in chickpea. So for that, I have gone for interolog mapping. So what I mean by interologs is, in an organism, if A is interacting with Y, and if A has got an ortholog in a different organism, so we say that X and Y are interologs. So X has an ortholog in X1 in an organism B, but X is interacting with Y in an organism A. So that means X and Y are interologs in an organism of our interest. So that way, interolog mapping is a basic step for deriving global protein networks, protein interaction networks, and often for building a protein network or a gene network, we would like to go for gene co-expression, co-localization, and functional categories of proteins. So ultimately leading to a protein interaction network, which would further help to identify some of the markers or triggers for androgenesis. So I have selected around 25 genes for my study as query, some of them being GST, glutathione S-transferase, alcohol dehydrogenase, SERK, AGL15. So I've taken 25 genes from model crops, which are either directly or indirectly involved in androgenesis and somatic embryogenesis. Some of the genes are essential for development of the microspores, and some are directly involved in androgenesis. So this is the methodology I've used for interolog mapping, where the first step is to analyze the ESTs and then go for the subcellular location and studying the families of these genes involved. Again, we would go for back-to-back orthology. After sequence similarity such as using BLAST and FASTA, we are going for back-to-back orthology to check whether our FASTA and BLAST results are appropriate or not. So that would give us a double clarity and double ensurety that we have done well. Then the next is to go for interactions, protein interactions. I have employed 3 different tools to study the interactions, that is Gene Mania, Intact, and Bind. So with these three tools, I have extracted the protein interactions and then developed a protein interaction network with one of the successful queries. I don't want to go into the details of this. It is just a representation of the BLAST and FASTA results we have got. As I've highlighted here, SERK has got a coverage of 99% in Chickpea. It's got an ortholog. And going from both the tools, that is FASTA and BLAST, we have got SERK as a candidate, and which was given a score of 1 based on both the results. And another 2 genes, brassinosteroid insensitive gene and FTSH genes. These 2 have very good sequence coverage and similarity in Chickpea. And 3 more genes here, arabinogalactans, 1433C and 1433A. All of these have sequence similarity more than 80% and a coverage of more than 60%. So we have gone for sequence similarity now, and we have also gone for protein localization and PFAM orthologies. So now the next tool is to go for co-expression of these genes, and this is a Pearson correlation, where I have used a tool called GeneVestigator tool, and we had a list of proteins which are co-expressed, and genes under this circle or this area are supposed to be co-expressed. That is 1, 2, 3, which is cellulase synthase and the WRKY. So this also validates the study. And then finally reaching the consensus for all the tools that we have followed. We have gone for orthology, the families, the functional linkages, subcellular location, the interactants, and interologs. So each of these categories was given a score of 1 and 0 based on their success. So any score, the total reliability score of more than 3 was supposed to be a good criteria to select it as a query. Among these, SERK has got a maximum score of 6, followed by AGL15 and LTP, that is lipid transferase protein. So both of these have got a score of 5 and 5. So from our consensus, I would say that SERK2 is a candidate protein or a marker for androgenesis in chickpea because of a score of 6, followed by AGL15 and LTP. So since SERK is satisfying our criteria, I have built a protein interaction network with SERK as query. Sorry, this is not very clear, but yeah, this is the query SERK. This has been built using Cytoscape, and we can see that SERK is self-interacting. These arrows represent the direction of interaction, and SERK is self-interacting, and it is under the direct influence of SERK1, which is another protein belonging to the same family. And further, SERK1 has these many interactions with both physical interactions and direct interactions. So I would say that SERK is a good candidate to be selected as a marker for androgenesis in chickpea. And chickpea is also a good candidate for anther culture or androgenesis or anther culture experiments. However, it does not validate the results because it is just a matter of 1 or 2 genes among the 25 genes that we have selected. Had it been, say, around 10 to 15 genes which turned up as potential candidates, maybe we could have found more information about it. We could have gone for interactome studies. We could have gone for chemical binding or ligand bindings with those genes. But it's just one query that has turned up as a potential candidate. So further, we just didn't stop with in silico analysis. I wanted to go for a proteomic study. So I have extracted proteins from anthers before culture and in culture in chickpea, and have gone for two-dimensional electrophoresis. So why go for two-dimensional electrophoresis and why go for proteomic studies? Because proteins represent the dynamic changes in a cell, and it's a complement to genomic studies. So I have gone for two-dimensional electrophoresis, and it's a brief protocol of the extraction. It's a dry, cold extraction of grinding of anthers and subjecting it to acetone and hexane washes. And I've used 0.05 molar tris HCl and PVP as the dissolvent for proteins. And these proteins were subjected to Bradford assay to check the protein concentration. And two-dimensional electrophoresis was done using Biorad protocol. And the isoelectric points we have selected is 3 to 10 and the molecular weight of around 14 to 100 kilodaltons. So this is the two-dimensional electrophoresis result. Unfortunately, this is our culture sample. It has not given us any spots, and the most obvious reason for this is the lack of enough concentration of proteins in the sample. So we could not go for differential expression because had it given us a better spots or better result, I could have gone for a differential expression comparing the spots in these two gels, but it wasn't the case. So right now MALDI is not the choice. Since I haven't got a good result with this, MALDI is not a choice for sequencing of the spots and picking of the spots. So in an alternative, I found a cheaper way to study the proteins. So I have gone for a virtual two-dimensional gel. It is using JVIR gel, a tool which gives us a virtual two-dimensional gel where you input your EST sequences to it and it would generate the protein two-dimensional electrophoresis gel according to the ESTs that we have given. And here there were around more than 1,000 spots. So overlapping this spots onto our experimental gel is, it would just result in a crowded experiment. So what I have done is I have selected a region between 4 to 6 and molecular weight of around 3 to 70. So I have got around 596 spots in this region which are probably involved in androgenesis and essential for development of microspores. And among these, as we have seen earlier, the potential candidates 1433C and Arabinogalactans AGP and AGL. So these genes were, these proteins were potentially identified as candidates or markers for androgenesis in chickpea. So coming to pearl millet, I would bore you with a little bit of pearl millet. So as you know, pearl millet, it's a staple food in semi-arid tropics and India's largest producer. So what is the status of double haploids in pearl millets? It is usually used as a pollinator for wheat and oat to develop double haploids. But the problem here is, since it's being used as a pollinator, most of the haploids result from the receptor, that is a wheat or oat, and there is no protocol as such in pearl millet for double haploids, and very sporadic results have been obtained in a very, like this, say around 20-30 years back. So there are no protocols for it, and unfortunately it has been the least explored cereal. Though the most successful protocols exist for cereals, that is wheat, barley, rice, maize, most of the successful double haploid protocols exist for these cereals, but sorghum and millet have somehow fallen behind. So because of that background, we thought it would be an interesting work to just give a preliminary attempt at double haploids in pearl millet. So for pearl millet, I have used whole spikes for culture. I have selected 7 genotypes: 89111, XL51, 4201. These 7 genotypes I have used for the experiments, and the whole spike along with the flag leaf was collected and given a cold treatment of 7 days, and untreated spikes have also been used as culture. The sterilization was followed using mercury chloride, 0.1% mercury chloride for 15 to 20 minutes. The florets were directly cultured, as generally it's been reported that direct culture of anthers is not successful in pearl millet. So direct florets have been cultured on the semi-solid media. So these are the different media combinations that we have used, predominantly having a combination of 2,4-D NAA with other cytokinins, carnitine, zeatin, thiodio. Unfortunately, cold treatment has destroyed my cultures. Most of the anthers turned brown within a week. The anthers are supposed to emerge out of the florets, and they turned brown within a week. It was unresponsive. So I have gone ahead for my experiments with untreated inflorescence, and 8% sucrose were most favorable. But when 4% maltose was used as a secondary carbon source, I have got proembryoids and globular embryos in pearl millet. So the best medium for me was the basic CH media that I have used for chickpea also, and I've used 8% sucrose, 4% maltose, and 2,4-D and DAP. This was the best media where I've got proembryoids and globular embryos. So here, just a representation of the effect of genotypes and on the microspore viability. As I said earlier, the viability was very high among all the genotypes excepting 4201, but the divisions were observed in 841P3, XL51, ICMB, 93333. Here, as I've already said, 13.7%. And this is a comparison of the light conditions on the divisions and viability in both light and dark. This is 1.7 sucrose and 8% sucrose. There is a almost similar response, I would say. But however, they responded only in dark. Light wasn't very effective, but the cultures maintained in dark were more effective, but there is not much of a difference between the response when cultured on 1.7 sucrose or 8% sucrose. And here the viability and the divisions, as we have already seen, a very high response among XL51, 4201, 841P3, and almost 90+% of viability after 15 days in culture disease. So these are the photographs of my multicellular microspores. This is the multinucleate microspore. Actually, pearl millet doesn't give multinucleate microspores. It is usually multicellular. After 15 days, we would observe many multicellular microspores more than multinucleate. So it was you can even see the division of cellularization here in multicellular, and this is a proembryoid which I've got after 20 days in culture, 15 days in 8% sucrose, and another 10 to 15 days in 8% sucrose with maltose. So we've got proembryoids. So again, I have gone for a similar analysis like I have gone for chickpea, just to check for the proteins expressed in pearl millet. And for in silico studies, pearl millet was not a good candidate. And I would tell you the reason also, because we do not have full-fledged EST data for pearl millet. There is very little data from selected experiments which people have uploaded in NCBI. So as in chickpea, we do not have an entire EST database or a draft genome. So whatever input I would give to the tools, they would return only limited results because we don't have the EST database. So in silico analysis was absolutely futile in pearl millet. However, the protein concentration was good, and I could get good two-dimensional electrophoresis results. And this is a pre-cultured sample, and this is the 15 days in culture. And here it's an overlapping. You could see the round spots, which is actually an overlap of the virtual gel on the experimental gel, in order to identify the spots. As I said, it's a cheaper way of deciding the spots than going for MALDI. However, ultimately MALDI has to be done, but as an initial experiment, I have overlapped the virtual gel onto the experimental gel. And these are the genes which are exclusive to pre-culture, that is Matchbox and MYC and RAP proteins, and ABC transporter and alpha malase were exclusive to 15 days in culture. I mentioned Matchbox and RAB protein and MYC here because, as we have already seen, MADS is very important in attaining the induction ability, and it is very important for induction of androgenesis. So, presence of this Matchbox at pre-culture would justify the better results and better response in pearl millet compared to chickpea. And 11 proteins among the 25 we have seen as a query are involved in androgenesis in pearl millet with the limited amount of data that we have. So that would be ADH, AGL6, MADS and RAB. So I would say pearl millet is a much better candidate than chickpea, obviously because of the results. And further experimentation with pearl millet would, I think, definitely be successful in getting regeneration from the pro-embryos that we have got. So in summary, the best part of my thesis, I mean, I love my thesis, so best part of my thesis would be the in silico approach to study androgenesis, because it has not been done. It's a first attempt in legumes specifically. So the protein interaction network that we have built in chickpea could be applied to other processes also, like apomixes or any other process like embryogenesis. So this is not specific. The method that I have employed here, it's not specific. It could be applied to any other crop, any other method, any other process in the system. And it's a validation because we have not just gone for one tool. For each category, I have gone for 3 to 4 tools to validate my results. So it's a first time attempt, and it would definitely give us a chance to build a network for androgenesis in the future. So my ultimate aim would be to build a gene regulatory network for androgenesis specifically, where you could even identify the triggers or the interacting partners and build a complete gene regulatory network for androgenesis that should be available as a database to everybody. And if you would want to see whether your crop is a candidate or not, you would have to just give your ESTs or your genetic data to that tool, and it should be able to predict a network on its own. So that will be the most useful tool for double haploids or androgenesis in any crop. So thank you so much for bearing with me for almost 60 slides. And I would like to thank Nalini ma'am, because without her I am not here, and she has always given me a lot of encouragement, and she's been a constant support. And special mention to Dr. Prashant Survajalla. He has helped me a lot with bioinformatics, and now I'm a thorough bioinformatics fan. I would like to go further in it. And thank you to ICRISAT and LSU team, Rosanna ma'am, everybody for accepting me and giving me this chance. And also to JNTU, because I've registered there. And thank you to Shilpa and Sandhya, who have always shared, we have always shared our difficulties, cried, fought, and everything. So it's nothing, it's not a good journey without good friends. So thank you so much for listening. Any questions? Ma'am?
I'll say Sameera, you have done a really good job.
Thank you.
I mean, it was a very difficult topic. You remember when we started that. And of course, the work you have done in proteomics is also very useful. But say if you were, given a chance to work only on developing double haploids in another 3 years, what is that you would like to do? Because that is still,
Yes.
is the most important thing we want to achieve.
Sir, my approach would be definitely different because when I started doing it for the first 1.5 to 2 years, I was just trying to reproduce the protocol and find variations with it.
Very well.
So it is. I feel it's just a blind trial and error method. We do not have any idea of how we want to proceed. So because now we are looking into the proteomics and we are looking into the genetic basis, the idea is to study further in silico to identify any say chemicals or ligands which would interact with these genes and use them in the media. Like for example, I spoke about arabidopsis. So in pearl millet I have used arabidopsis and it was definitely helpful. So my approach would be definitely different. I would definitely love to develop double haploids in some crop. It would be my lifetime goal, but the approach would be different. It would be a systems biology approach. It should not be a single tissue culture approach or genetic approach. It should be a systems biology approach. That's what I think.
Yeah, I mean, it will be developed one day,
Yeah.
eventually, because,
Yeah.
every cell is 20%, so it has to be, but, we are not able to crack it, but it will be done.
Yeah.
So,
I think systems biology is what we have to do, sir. I mean, we have to look into other criteria, just maybe the trigger and all. We have to look into that instead of just going blindly for tissue culture. Maybe incorporate arabidopsis or steroids into the media. So that way it would be different. And even microspore culture has to be explored. Because I have mostly worked on Anther culture. Microspore culture I have done just a few, but I wasn't very keen on that. So microspore culture has to be tried, which you can
Thanks, Sameera. You have covered a wide range of aspects for the study. I'm just wondering, I have a sequence of questions, so let me put the first one now. How many anthers have you cultured in each treatment?
That would be around 30 florets, ma'am. 30, that is 30 and 10 anthers each, so 300.
300. So when you are culturing only 30 florets of one genotype, do you think this is really something very great efforts which will respond? Because 30 florets, another thing is you yourself mentioned about the genotypic specificity, another is seasonal variation which is critical.
Yes.
So in how many seasons did you conduct the study?
I haven't taken field samples, ma'am. Mostly I have taken from the glasshouse. So glasshouse anyway it is controlled facility. So for me there will not be a lot of seasonal effect. But yeah.
No, another thing is in which season or in which month did you collect the florets?
I collected throughout the year, ma'am, because I have used them from glasshouse.
No, in glass house also, because I'm growing chickpea over there, but when we take the florets or even the pot set, in the cultivated ones, it gets very low when the flowering comes in April or May.
Yes.
So definitely, even within the plant, the pot set is not there, but when we are taking the anthers and culturing, and that too only 30 florets per genotype. Another thing is at what stage you are taking them, whether in the beginning of the flowering, in the middle of the flowering, or at the later stage of the
So
flowering.
that, ma'am, the first 2 flushes would be the best, ma'am. After the second flush, the quality reduces. So the first 2 flushes are very good.
And another thing, you showed us a slide where can I see the auxin and your cytokine concentration in the beginning itself? Yes. So what was the criteria to select these doses? If I see in the slide, your auxin concentration is really very high.
Yeah.
So I think that could be one of the reasons that you are getting the multicellular structure and it is not through the direct Androgenesis. So what was the basis for selecting this
No, the basis was there was protocol. The concentration was maintained, but the hormones were changed. I have used different hormones, but the concentrations were the same. The basis was the CH1 medium, RMD medium by Greville's protocol. Because when I started, as I said, it was only able to reproduce the protocol and try variations. So that was the basis of the experiment.
But you didn't try by increasing the cytokinins. And you could have tried other way. Because if you see here, oxygens are that much high, in any case, it will not give you direct organogenesis, it will lead to the callus formation. And that's what you have observed in your study.
Yes.
Now, another question is, as you told about the candidate gene studies, 25 candidate genes, did you try to correlate these things with rice and brassicas? Because these are the two crops.
The genes were selected from monocrops only, that is barley, rice and maize. From all the studies that have been done till now, based on transcriptome or proteome studies that have been done, that was the picture that was there. It was published in 2005 by Marashchan. So from that, I have added few more genes to that. It is not just a direct copy. I have updated few more genes from recent studies in 2013 and 14. So from all the model crops, whatever genes they have selected as markers at different stages, that I have selected, and among them, 25 most important ones which are validated by different people. So not just one person, so different people have validated those 25 queries. So those have used.
So did you find anything which is there in rice and brassicas? Because brassica is something which is readily responsive and which is also present in chickpea, or something which is present in rice and brassicas but not in chickpea?
I would go to that slide one because I'll just show you. Yeah. As you see here, I have mentioned Arabidopsis here because it is the model crop, but not that it is the source from that crop. However, you can see here SERK is one, then cysteine precursor from maize, then phytoxin precursor from Mollium Valvaire, Brassica, that is NAP. You can see napin. Yeah, napin, NAP, that is NAP is a seed storage protein, that is from Brassica, and it is also present in chickpea. Yeah, because I got a medical ortholog for it, so it is also present in chickpea. Even AGL15, AGL16 and WRKY genes, those are all present in chickpea. So yes.
So if they are present in chickpea, so what do you think? We need to go for intensive efforts because you have cultured only 30 florets, 20 to 30 florets.
30 is like the data, like basic data, but more than that I have tried, I might like...
No, because anther culture is something where really, initially even those wheat and other crops, they were, recalcitrant ones.
Yeah.
So recalcitrant means we are not able to find the switch.
Yeah.
But it's not like only her work, if you see like others also, they have been working continuously, they have captured so much they have done. There is still something which has to be there. It's not like, okay, she has done only few, but we know that other labs also have worked so much. So there has to be something, there is something still missing. So even if she has done more, she would have got the same result because it's not the number game in this case. There is something missing. Because it's not only her, there are other labs they have been doing. Nobody is getting. Even they are not able to repeat their own protocol. They are not able to do it.
But I wanted to check because I remember you talked with Greva, isn't it?
Yes.
Yeah. And what did she say to you?
No. Actually, at that time that was only through email. So they were quite confident that the protocol is good. And at that time we were just trying to explore the possibility of getting seeds from them so that we can use the same genotypes and see whether the protocol is working or not. But after that, actually, the study was not carried out further.
That's why we sent her to Australia, the lab, they still, not able to repeat it. So it's not, I still don't believe it's number game. It has to be, it has to be something. Still. There is something missing. It will be done. Somebody will do it. Somebody will do it for you.
But also, whenever I don't know, because whenever you are modifying the media or like that you did by several factors, that some of these really basic things, that whether you include the greater proportion of genes or cytokinins and like that, perhaps that could have been taken care of, so that you at least, because you are modifying certain things, and if you really put something which is not likely to work, then your whole efforts So somebody should have guided you perhaps more appropriately and like that. Another, very small thing I was wondering, you have written some there 8.89% sucrose.
Yes.
So why 8.89 and somewhere you write 8? So why don't write these straight things whether 8.5% or 17% or is there something very...
No, it's just maybe my overlooking, but it is 8.89% sucrose and 17%.
I'm asking that why 8.89? Why not
8.89 because that was used for microspore culture in chickpea by Monica and Janine Crosser.
Okay.
They have used it for the microspore culture.
8.89.
Yes, sir, 8.89.
I don't know wondering. Simply if it is 17% people go 8.5%, 17%, 25.5%, that kind of thing then people do.
So, thank you, Dr. Kiran Sharma, sir. And he wants to ask one question. What are your recommendations for chickpea haploidy work in the future?
Ma'am, as I said when sir asked, I would definitely suggest going into further proteomic studies or at least cDNA transcriptomic studies in culture a lot and go for pre-cultured samples and check for the cDNA, check for the proteomic studies and then decide whether there are certain genes which you could trigger. So triggering those genes, presence of those genes and triggering them is important. So first of all we need to know whether these genes are present in chickpea or not. And as we have seen some basic studies that have done, we have seen that there are some candidates. So now it's the next step to identify triggers for these genes. So if we identify triggers, maybe they could be amino acids or simple carbohydrates or simple components like chemical, ligands or metals, maybe we could include those in the media. So that is why I am emphasizing on the systems biology approach. The next approach definitely is to identify triggers and then include them in the media. So maybe that would be definitely helpful. Yes.
In commercial plant breeding, double haploid is successfully used in maize.
Yes, sir.
So that is a different technology, not Androgenesis. We cross with the tester, and that technology is also not genotypic specific. So successfully used. So how do you think those types of technologies have some probability of success in these crops?
So permanent, yeah, maybe that way it could be tried because since it's also a cereal. But chickpea, it would again you would have to sit in the field or in the lab and then cross and do all the study. So I think if double haploid through Anther culture or microspore worked out in chickpea, then that would be the best. So this is a faster method. As you said, crossing and all.
But your panel technology should not be genotypic specific.
Yeah, it should not be genotypic specific, definitely. Definitely shouldn't be.
Sameera, that was really good and wonderful presentation. I have two questions. I'll quickly come out with them. The first one is, you said you are trying to look for the co-expression and co-localization of different proteins. So, because you were using a Zeiss fluorescent microscope in the lab, did you try using different ELISA flow dyes to see where exactly you have, what do you say, to visualize the nuclear divisions in a cell? Did you try? Because you have shown the DAPI pictures, but did you try exclusively for the nuclear targeting dyes?
No, DAPI specifically stays nuclear.
Yeah.
So DAPI is good enough.
The green filter and the red filter, laser flow dyes you get, so where you can exactly see the demarcate between the cytoplasm, nuclei and...
But even here you can demarcate, you could clearly see the two nuclei. DAPI stays specifically to the nuclei. Maybe you would, as you said, it would give us more clear picture.
Okay then, I'll talk to you. Another thing is, you said protein-protein interaction study. Did you go for pull-down assays?
See, the pull-down assays come after you go for in silico studies. So that is much more advanced. Without knowing what proteins are there, without knowing what interactions are there, if I just go for antibodies and go for immunoprecipitation, I would be spending so much time. Money and time is waste. So I am first looking into the in silico techniques. So if I have good results here, then I would definitely go for pull down.
So that was a crude thing. You never went for purification? It is not at all an wet lab experiment, purely dry lab experiment. So since we are seeing that SERK and AGL are good candidates, now the next thing would be to have an antibody for SERK or AGL and then go for pull down assays, or these two hybridization.
So the co-localization thing is also a dry
It is also a dry thing, yeah.
Okay.
That is...
so you never visualize co-localization in a cell?
No.
Okay.
That would be the next step.
Okay.
First validation has to be done.
So let's give a big round of applause because she has really done